Proteins are polymers whose complex chemistry and physics, in the course of evolution, permitted their recruitment for immensely diverse functions in the biological machinery. The study of protein conformational behavior and interactions of proteins with other molecules in the intra- and extracellular milieu are of great interest. One of the biggest challenges in understanding proteins is getting sufficient spatial and temporal resolution in measurements. For example, consider the fluctuations around a protein binding site as a ligand interacts with it. Currently, we rely on molecular dynamic simulations to move from the static structure obtained from NMR or X-ray crystallography. But how can we advance in capturing these dynamics experimentally? Recently, scientists have pioneered creating and applying site-specific probes to increase structural resolution. When coupled to a spectroscopic technique with time resolution in the picoseconds, we can get closer to the ideal scenario of studying proteins atomistically. In the perspective we wrote, we focused on vibrational probes, specifically those containing nitrile, azide, cyanate, and thiocyanate moieties in proteins and nucleic acids. There are several requirements to become a candidate as a probe molecule. The vibrational transition must reside in an uncongested region of the spectrum. It ideally will have an extinction coefficient comparable to or larger than the amide 1 mode of peptides, and it must be sensitive to its environment. For an example, we show a binding experiment between chemodulin and the peptide MLCK, which binds chemodulin with nanomolar affinity. Here, a nitrile derivatized amino acid, para-cyanophenylalanine, was inserted into the MLCK sequence at a position that is buried in the bound complex. As you can see, there is a peak frequency shift from where the probe is solvent exposed to when it is buried in the complex, which is due to a combination of several factors discussed in detail within our perspective. There is also a change in the bandwidth of the primary peak upon binding, which contains additional information regarding the system of interest. Additionally, we present two examples of applying vibrational probes to better understand the local environment of proteins. A major advantage of these probes that has become increasingly utilized is their ability to measure the local electric field, especially in binding site regions where the field is dynamically changing depending on whether a ligand is present or not. Another method is to use these probes in conjunction with attenuated total reflectance techniques to site-specifically measure transition dipole orientations with respect to both the membrane normal axis and helical axis of a membrane-binding peptide. This could be extended further to even obtain structural constraints for solution structures of these peptides. Over the past 10 years, there has been an explosion of vibrational probe development and application as tabulated here. All of the probes presented meet the requirements we mentioned earlier, and each has strengths and weaknesses associated with its use that makes a choice of vibrational probe context-dependent. What is the nature of the sensitivity of these probes to their local environment, however? To understand this, we turn to computational modeling. The development of experimental probes has occurred almost simultaneously with the emergence of computational methods capable of accurately predicting vibrational spectra. Modeling the vibrational spectra of probe molecules greatly increases the amount of information that can be obtained from a given experiment. For example, employing a combined molecular mechanics quantum mechanics approach, we investigated what interactions between water and 5-cyanoindole give rise to its nitrile stretching spectrum. Specifically, we showed that the bandwidth of the nitrile stretching band of 5-cyanoindole is not only affected by interactions of water with the nitrile group, but also by interactions of water molecules with the pi-electron system of the aromatic indole ring. Therefore, this study provides a molecular basis for understanding the hydration-induced broadening of the nitrile stretching band of this compound. In the future, related approaches will facilitate the interpretation of experimental spectra of probes immersed in the complex and heterogeneous environments of protein matrices.
There are several other applications of vibrational probes that we touch on in our perspective. We expect that the number and variety will continue to increase as researchers move away from probe development and towards asking specific biological and physical questions of systems of interest.
We conclude with some comments on what we expect the future holds. We've seen linear studies aimed at understanding the local dynamics and heterogeneity of proteins of interest, but nonlinear methods such as 2D-IR will allow us to differentiate between different types of spectral broadening to understand the nature of structural motions and give us access to a time regime to catch the fastest structural fluctuations. Additionally, we do expect that there will still be some development of new probes in the future, despite the large list that has already accumulated. Furthermore, computational modeling will continue to develop, as well as experimental efforts to look at water dynamics at solvent-protein interfaces. One exciting area of study will be investigation into coupling between these different probes, whether via resonance energy transfer or not. To test this, we've created a tripeptide containing both p-azidophenylalanine and p-cyanophenylalanine. We hope that between this video and our recent publication that we've convinced you of the utility of these reporters and given you a look at the history and current state of the field. We'd like to thank the Journal of Physical Chemistry Letters for the opportunity to create this video, as well as the National Institute of Health for funding.